The overall goal of this procedure is to map mechanical properties of plant tissues using an atomic force microscope. First, prepare glass slides by pipetting a droplet of low melting temperature agarose onto an etched region of the slide to form a thin agarose film. The second step is to dissect and mount the sample. Remove floral buds from the stem, cut the meristem away from the stem, and immediately embed the cut meristem in the thin agarose film on the glass slide. Next, add enough melted mounting agarose to fill in the crack created when the meristem was embedded in the agarose film. The final step is to set up the atomic force microscope for data acquisition. Ultimately, data analysis and measurements of the apparent Young's modulus of cell walls generate a mechanical map of the properties of cell walls simultaneously at subcellular resolutions and across entire regions of tissue.
The main advantage of this technique over existing method is that it can measure mechanical properties of the cell wall simultaneously at the subcellular resolution and over entire region of the tissue.
The agarose media for embedding samples is prepared from 0.7% low melting temperature agarose in 10% mannitol in water. Using a strong metal instrument, such as a drill tip, etch out a 0.5 by 0.5 cm area in the center of a microscopy glass slide. This roughens the surface in order to facilitate the adhesion of the agarose to ensure that the agarose media sticks or fixes to the slide. Pipette a droplet of about 20 microliters of embedding agarose media onto the etched region of the glass slide. This produces a thin film of agarose. Store the glass slide.